Hi everyone, welcome. Bonjour à tout le monde. We're going to wait just a few minutes for everyone to get connected and then we'll get started. Okay, so I guess we can start and we will welcome people as they join us later on. So welcome everyone to this webinar. My name is Julie Champagne. I'm a researcher with the Energy, Mining and Environment Research Center at National Research Council. And I'm also vice chair with the Environmental and Social Responsibility Society, ESRS. ESRS is one of the many volunteer societies of CIM. ESRS's vision is to provide leadership and professional development opportunities as they relate to the social and environmental responsibility and performance of the Canadian mining industry. Webinars are one of the ways we promote discussion and share leading practice on current environmental and social issues. So please contact us if you want to be more involved with ESRS. Our website is accessible through the CIM website under the Communities tab. ESRS also promotes diversity and inclusion and works to raise awareness on Indigenous issues within the mining industry, where natural resources, land, and nature are at the heart of all of the activities. So I would like to take this opportunity to acknowledge that myself and others who are based in Montreal are standing upon unceded Indigenous lands, where the Mohawk Nation, Kenyankeha, is the custodian of the lands and the waters. And if you don't know about the Indigenous nations and cultures where you are based, we encourage you to look it up and to share around you. If you do know the information, you're welcome to share it in the chat box. During the webinar, we would like to ask you to write your questions in the question box, not the chat box, and they will be addressed at the end of the presentation. So now let me introduce our speaker for today, Dr. Aria Han. Aria is passionate about data and believes that combined with intelligent, statistical, and computational technologies, data hold the answers to many burgeoning questions facing multiple industries today. Aria has built tech to solve technical challenges for multiple industries and is now helping to scale technology for microbes in mining through Kunki Canada, a bioinformatics company that she helped found and grow. Aria's strengths are in her abilities to work effectively with multiple stakeholders to take large-scale ideas from concept to reality and create knowledge translation materials suitable for expert and lay audiences. Over to you, Aria.
Thank you very much, and thanks for having me. I can't see anybody for the record, so if something stops working, you can't see me or the presentation, please put it in the chat or questions so that we can make sure that that gets fixed as soon as possible. Really happy to be here. Thank you for the invite. I really appreciate it. It's always fun to talk about this topic, which is definitely close to my heart. The title comes from something actually a sales manager for a microbial products company told me the day before he retired. He said, microbes may save us all, but my work is done. And that line has really stuck with me, and I like it. I gave a similar presentation to a slightly different mining audience a couple months ago. And at the end of it, someone came up to me and said, be careful where you give that presentation, because they're going to burn you as a witch in other mining environments. So with that, I am going to launch right in and fearlessly show you many of the things that I showed that audience, and then I await the comments and questions about how crazy it actually is. I always like to start here with microbes, to say that when most people hear microbes, they think of germs. You think of sharing a cup or something that makes you sick. I mean, COVID's been a pretty big thing. It's like definitely on our radar in terms of germs, but microbes are so much more. Science is a way to have new thoughts about the world. That's one of my absolutely favorite definitions of science, and because it's simple. It is just a way to have new thoughts about the world, and that's really the goal of today, is for everybody to come away from this talk having new thoughts about the world and a new understanding about what microbes are and how they might actually be playing a bigger role in your current life and our future than ever before. So I've got a couple of beautiful pictures of microbes up here. They come in all shapes and sizes, and they are found absolutely everywhere. I love this slide. I use it often with all sorts of audiences, because I think that the feeling of when you're camping or you're out somewhere and it's dark and there's no light pollution, and those areas also tend to be really quiet. So there's this like darkness and this quiet and this calmness, and you're looking up at the stars in the night sky and you can really see them, that we get this feeling that I think is pretty universal, of feeling really small in all of that space, and with the vastness of the number of stars is really overwhelming. And if that is a feeling that that resonates with you and that you've had, I just want to put this all into perspective. There are 10 to the 22 stars in the known universe. That's all of the stars in the known universe, many more than you can see on that cold, dark night when you're camping. But there are 10 to the 30, so 8 orders of magnitude more microbes on planet Earth than stars in the known universe. And so if you get that feeling of smallness and this vastness when you're looking at the night sky, you should also get that feeling, and perhaps amplified by 8 orders of magnitude, when you think about just the absolute overwhelming number of bacteria that exist on this planet. Microbes get this bad rap, right? We only talk about them when they're sick or they're doing something. It's like, oh, you can't get spinach anymore, there's some kind of outbreak. But that's not how they work. And I think this has been the way that microbes work has actually been really hidden. So we hear about them when it is a single species. There's E. coli, there's an outbreak, there's salmonella, don't touch snakes, and then, eat food, which is like a weird thing to tell people because I don't know how many people are handling snakes all the time, but that's like, in my surveys, one of the things that comes up, which is true. I mean, wash your hands, but like you should probably wash your hands before you eat, regardless of like handling snakes or not. Microbes very, very rarely actually live in isolation. They live in communities. And so when we hear about a microbe, a species, it gives this impression that there are these like single entities that are wreaking havoc or doing all this work on the planet. And the reality is, no, they live in these really, really complex communities actually, or networks. And so they are signals that are being passed through microbes all the time. You're contributing to this. So every time you're talking to somebody, you're close to somebody, physically, doesn't count on Zoom, you're not changing cells, you are part of passing information among microbes. So you're a part of the microbial network, and you're a vector for them in transferring information and bringing them all over the world. Every time you travel, talk, interact, buy something from the grocery store, any interaction you're having with your environment, you're actually a part of these microbial networks and information exchanges. To put it into perspective, you have about 3 trillion human cells. It sounds like a lot, and like, great, good for you. You're multicellular, that's absolutely fantastic. Microbes are single cellular. Every entity is just one single cell. But you actually have, depending on the time of day, if you don't get this reference, ask the question and I'll answer it later, but do know that the answer is slightly gross. Depending on the time of day, you have 2 to 10 times more microbial cells in and on your body than human cells. So by cell count, you are more microbe than human, by far. So, more microbe than human, you have all these more cells, you have more microbial genes than human genes. We think humans have about 23,000 genes. Most of your DNA is not even in genes. People are like, oh, genes are the way that it goes. No, the genes you have as a human are important. Obviously, they make you, but the genes, when you hear comparisons, actually comparing how similar humans are to bonobos, which is our closest relative, is actually kind of complicated to do well. But we say, we're 90, 95%. Some people would even say up to 98% similar to a bonobo. And you're like, okay, but I still feel pretty different from a bonobo, and you should. But it turns out that actually the similarities between you and the bonobo, or you and the chimp, or you and any other kind of mammal, as they get more distance, it's actually in the genes. It's not in between. It's the things that are in between the genes that are more different. And if you think about it, it makes sense, because genes are these building blocks. So really, you're going to see this as a theme. I'm going to talk about DNA as tools and building blocks, and with a lot of construction analogies, which is out of character, but actually just kind of fits well. Genes are these building blocks. And so if you think of your parts, like your hair, your skin, your liver, your heart, and you think of a bonobo's parts, they have bones and ribs, and generally we got the same organization, very humanoid shapes. It's really, you have all the same pieces there. It's just how they've been put together is slightly different. And that's largely why insulin is insulin, hair is hair, those proteins are proteins. But how they're put together really makes a difference between you and a bonobo. And then between other humans, you could be 99.9% identical across your genome to people that you're not even related to. So, we're all the same species. It's all very similar. And there are these slight differences that make us very different from one another. And microbes, again, you're more microbial than humans. So they're affecting, it depends which literature you're reading right now, but microbes have been shown to even affect the behaviors in mice. So it could be that some of your behaviors have been microbially driven, but definitely they're helping you with your digestion. You can't do a whole lot without them in your stomach. They're helping you with your skin, your general maintenance, and you want to be packed with all of these bacteria, because if the good bacteria, and this is like good and bad, is like, it is what it is, but the good bacteria, I'm just going to define very broadly and non-specifically as bacteria that don't make you sick, and bad bacteria is bacteria that make you sick. So if we take this very simple approach to good and bad, you want to be packed full of good bacteria, so there's no space for the bad bacteria to get in there and take over. COVID has been horrible, and we can talk about silver linings, but we're still really feeling the effects, and there are countries that still do not have adequate access to the vaccines and still have really horrific COVID outbreaks that are affecting them. But COVID is a good example of the whole point of information exchange among people. So it is this really beautiful illustration of a new virus popped up somewhere in Asia and has spread very quickly across the whole world, person to person. And so if you're skeptical about this concept that you're really just, living your life, but ultimately just passing information from microbe to microbe to microbe, you're just one of their worker bees, this is a great illustration of how true that is, and how much how quickly and how much information can be exchanged across the entire world just through your day-to-day activities. And so we look at this, we think, okay, so there are so many microbes on this planet. They are incredibly diverse. And for most of them, we can't culture them, which means we can't grow them in a lab. So I think when I say I'm a microbiologist, people kind of expect microscopes and petri dishes. Never touched either one of those things. There are, and the estimates are hard to know because we haven't fully mapped the diversity of microbes that live on this planet. But generously, we can grow in a lab about 10% of microbes, which means 90% of microbes, we can't grow them. There's a whole host of reasons, and I'll give you I'll give you a couple of the reasons here, but you know, there are more. But if you think about yourself, and so you think about, let's imagine there was an alien species. See, there's this whole space thing here going too. It's all going to connect, I promise. Follow me, it's all going to connect. Imagine that an alien species is studying humans on Earth, and so they're observing Earth from, I don't know, they're very giant, from outer space. We don't even know they're there, and they're watching us, and they're like, okay, I want to study them more. I want to essentially culture a human. I want to grow a human in this tank so that I can learn more about it. But first I got to know, like, you know, how do I create those growth conditions for them so that that human doesn't die when I pick him or her off of the Earth and I put him in this little cage. It's like, okay, got the atmosphere down. They do some research and they're like, yeah, this group of people is eating, this doesn't have to be your diet, it's just an example. They're eating like a lot of nachos. And you know, so that's corn and cheese, which is a milk product, which comes from cows, and they're putting ground beef on these things. Fantastic. And so into this cage, you have the right atmosphere. And so you're scared because you're in an alien cage, but also you can breathe, so you're totally alive. And then in drops a cow and some corn. And you're looking at it and you're thinking, like, what am I going to do with that? Because I have to grow that corn or process that corn, grind the corn, cook the corn. Like, what am I going to do with that? And even if you had access to electricity and an oven and a grinder, you've got a live cow in there. And so even if you could milk a cow, which is a big if, because not everyone has that skill, you get some milk, but turning that milk into cheese takes some expertise and equipment. Turning that cow into something that you could eat in terms of beef onto nachos is, you know, a lot of steps in there. And you don't have those tools. And even if you had those tools, you can't butcher a cow in a closed space and then not die of infection later. What are you going to do with your own wastes? And so, you know, and the problems could, you know, continue to blow up. You can see where this is going. The same exact thing happens to microbes. They are used to living in communities. They're used to living with each other. They're used to others being able to process their food into a form that they can consume. They're used to others picking up their waste and removing it from them. They also fight, so there's chemical warfare that happens between microbes. They truly live in these very complex communities. And until we understand all of the elements that really need to go into creating a sustainable ecosystem for them, we can't grow them. And even more, it's very unlikely that even if we do understand all of those things, that we'll be able to grow each individual microbe in isolation, because again, they just don't work like that. They, you know, it would be very convenient for us, but nature really doesn't care what's convenient for us. So instead, we study microbes based on their data. And so we study microbes based on their DNA. And as you can imagine, this is the number of microbial cells. They all have their own genomes. No, we're not looking at the genomes of all of these. We obviously can't go and sample every single microbial cell. We're sampling communities at a time. But even if you're studying a couple of communities, it's big data. And so when we talk about microbiology today, instead of thinking of microscopes and petri dishes, think about supercomputers and rooms of servers like this, because this is a more accurate representation of how we're studying microbial diversity today than microscopes and petri dishes. It's funny, I was talking to a friend recently, we were talking about, you know, strange definitions of what's natural and unnatural. And those are actually really hard questions to answer because at some point, everything was natural. It had to come from the planet. And microbes are involved in all of them. And so when you are looking at the night sky, northern lights, and forests. Those forests are being maintained by microbes who are taking nitrogen and fixing it so that the trees can grow. They're degrading the leaves and needles that fall into the soil to create the carbon and phosphorus and sulfur elements that the trees need to grow. The animals that eat the trees that are then consumed by the larger predator animals, have those predators have microbes in their guts that are allowing them to digest this. There are absolutely, there's microbes everywhere we've looked, from rainforests to underwater to the far north. Kunki is a bioinformatics company, and so we look at DNA data from microbes from around the world. And we have worked with NASA to look at several extreme environments. And so when I say they're everywhere, I mean everywhere. We're finding them at the deepest depths of the ocean. We're finding them in Antarctica. We're finding them in closed lakes in Antarctica. The soils in Antarctica are really shallow because because it's been covered in ice so recently, it hasn't had millions of years to wear that rock. And so the soils in Antarctica can be 20 centimeters deep, so quite shallow, and they're dry, and they're still kind of rocky, and you'll find microbes in there, and you'll find that the microbes are different near the top than at the bottom 20 centimeters. It's all they live in hot springs, so they live in Hawaiian latitudes. There's other places we've gotten pretty beautiful microbial data from. So microbes are really living everywhere we're looking. And NASA's interest in that is to understand the limits of life. So what are the limits of life on Earth? And from that, what can we learn about where life may be outside of this planet? We're learning about the limits of life because we want to know, as our world changes, what is going to live there. And so some of these extreme environments on Earth give us really good analogs or comparisons to things that are happening in outer space, but they also give us good ideas on how we might adapt chemical processes, what biology might exist as our planet continues to change. So they're everywhere. There's a lot of them. They're dominating everything. We're studying them with their DNA. So you've probably seen this image or something similar. It's this double helix. We shouldn't forget Marie Curie's contribution to this as well, which often gets set aside, but that record has been set straight. And you've probably been taught that DNA is like a blueprint. This is a blueprint. You can tell what this is. And if this was a live audience, I would say, what is this? And people would say it's a house, hesitantly, because they think it's a trick question, but it's not. It's just a house. But from this, you can tell how many doors this thing has, where the chimney is, where the windows are. If it was a clear enough image, you could get a good idea of scale. You could measure where the roof is. You know a lot about this house from a blueprint. When you get DNA data, but that is not, so I hate this analogy. I hate this analogy because it's not how it works. When you get DNA data, you have no clue what it is. So there's not a person on this planet who you could give them DNA data, A, T, C's, and G's. Remember, there's only 4 letters, all the way back to like grade 10 biology. There's just A, T, C's, and G's that make up this double helix that's the DNA and codes for everything in life. And the most common question I get at this point is, well, if there's only 4 letters, why isn't this a solved problem? And then I say, there's a limited number of musical notes, there's a limited number of letters in the alphabet, and there is an untold number of stories and songs and poems that we can create. Same thing with DNA. Look around, everything that's alive, that's got DNA. There seems to be no limit. Things are evolving all the time. But even if you look at those A, T, C's, and G's of an organism, without being able to compare it to something, you have absolutely no clue as to what it is. We used to think that the more DNA an organism had, the more complex it was. And then we discovered that the onion has way more DNA than a human. So that kind of went out the window. I haven't heard a good argument for why onions are much more complex than people, but I'm open to it. So generally now we say no, genome size and complexity actually are pretty unrelated, which was surprising. You'd think like, well, I'm complicated and I should have more. And it's like, well, an onion has a lot of layers, so do with that what you will, but less complicated. So instead, I want you to think about genes as tools. So I told you more construction things coming. So we get these A, T, C's, and G's, And we need to do these comparisons to figure out what those tools are and do. And so you can imagine there's only 4 letters. These are really big files. Your genome, for example, has about 3.2 billion ATCs and Gs in it. Microbes have way less. Their genomes are a lot smaller. It's like, thousands, hundreds of thousands, millions maybe, but definitely not in the billions. Much, much smaller. And we're always looking for these small changes. And so if you think about it, I said, you're probably 99.9% similar to lots of people that you're not even related to. So that's not even talking about like your sister. It's just because you're human. And so the changes between you and me are small. Did you see it? So we're doing these comparisons to try to find really small changes here. And small changes can have big impacts. And so if you look at the pick, because this is mining, so old school pick is my example here. If you look at the pick in the middle of the screen, the length of the handle changes slightly. So we are looking for tiny changes in the DNA. We're looking for an A to become a T or a C to become a G. We're looking for these tiny changes in the DNA to discover, oh, when you make these small changes, the tool changes. When that tool changes, you can do different things with it. So genes are really tools, but to understand them, we need to do these large-scale comparisons. The more data we have, the better our comparisons can be, the better we can look for these small changes. Everything really comes down to having a lot of data. So this is Moore's Law, which you're all, I'm sure, familiar with. But what I'm really trying to show you here is that we can do this today in ways that we absolutely could not have done 10 or 20 years ago, when it was just absolutely, completely unaffordable to get the DNA that we need and the amount of data that we need to start to really look at these things. To put it into a human perspective, the first human genome cost about $3.2 billion to sequence, so about $1 per base, per like ATC or G. And today we could make a comparable genome for about $1,000. And so from 3.2 billion down to 1,000, you can imagine, like, yeah, now it's much more affordable and we can do it. And so now is our time. Now we can go in and look at that vast diversity of microbes. And so we call this the matrix. We're like, this is the real matrix. The matrix is the DNA and information from all of these microbes that are on the planet and maintaining our ecosystems for us and making sure that we are alive. So like I said, microbes don't live as individuals. They live in populations, and those populations live in communities. And it's at this community level that we get emergent properties. So the sum is greater than the parts is absolutely well applied to microbes. They are making sure that our atmosphere remains breathable, that things that we have, that CO2 is processed through trees that require microbes that actually like live within the phyllosphere, so like within the leaves of the microbe. Microbes are associated with the tree roots, and trees and most plants would not survive at all without microbial communities. The water that you drink is cleaned by microbes. Like nothing on this planet works without microbes. You take away microbes, we all die. And so there are lots of questions that we have, because this access to the information, that volume of DNA of microbes that we need, is just now available to us. And so we're actively developing the tools and computational infrastructures and reference databases to do those comparisons to every day. And so there are big computational problems. We're working in industries like wine. We're asking questions about what small changes in the genome of a yeast species makes this wine taste absolutely fantastic and this wine horrible. We are doing research in Antarctica to inform places in Mars to see if there are similar environments in Antarctica that look like what we think it looks like in Mars. And so are there microbes that live there? And if they do, does that mean it's possible that there's life on Mars, or that there was life on Mars? We're looking at agricultural sites. We're trying to see how can we improve the yields of plants. We've done research into babies. This is actually like a little bit of a sad story. There's a big Canada-wide study on baby poop. And the idea is you look at the poop, you see the microbes that live in the poop, and then From that, you can tell the microbes that are living in the stomach of the babies. And we wanted to learn how do the microbes in the stomach of babies, is that a good predictor of their health as they age? And so we had data from birth, 6 months, 1 year, 2 years, and 5 years from a whole cohort of babies across the country. And the answer actually, we did all the fancy math and analyses, and we got this model that said, if you know the microbial community of a baby at 6 months or a year, we can tell you with, a fair bit of statistical power, the likelihood that that child is going to have allergies or asthma as they age to 2 or 5. So, and so we were just absolutely, we were thrilled with this result. And then we looked a little bit harder, and what is an equally good or better predictor of that is actually socioeconomic status of the parents. And so that early environment for babies really sets up that child for success and health in a lot of ways. It's not the only factor, absolutely, but lower socioeconomic status on the coasts, for example, often is associated with living in basement suites or places that have poor ventilation or dehumidifiers. And so that increased moisture in those very young years is believed to be one of the causes of the colonization of microbes and fungi in the gut that aren't actually beneficial. And then, of course, we're doing a lot of work in mining, which I'm going to spend a lot more talking about. The problem here is the world's on fire. We all know it. Climate change is coming for us. I had this very upsetting talk over dinner with an older scientist recently, and he said to me, I used to think that the major effects of climate change were going to be felt after my death, and now I think that I'm going to feel them too. So the stakes are high. We have a lot of problems to solve. Climate change is definitely coming for us. There's, I heard a climate scientist say, we've already committed to feeling the consequences of some of our past choices in terms of climate change. And so it's there. And every interaction that I've had with mining companies to date has been one of genuine curiosity and wanting to do things in a more sustainable and greener way and solve some of the problems of the past. So the stakes are high. We have a lot of things to work on here. There's a lot of problems and challenges, but I think that microbes might be the solution. We already know that microbes can do a lot of the jobs that we might want them to do. So there are microbes that can eat radiation. And so, how do we expand these technologies and develop technologies with these bacteria to really solve some of the radiation problems? There are therapeutics that are coming out for people with type 2 diabetes, for C. difficile infections in your intestines. And so we're starting to see some of these microbial technologies come out and be products. There are lots of products that are microbially based in agriculture. They can either promote growth and act as like biofertilizers. And there are also microbial products in agriculture that can help decrease the amount of viral infections or blights, things like that in products. And we also know that microbes can be used to liquefy metals and get the metals in solution. And from that solution, we can then start to do biometallurgy. So there's big opportunity here. There's big opportunity in mining. The world needs mining. Kind of preaching to the choir in this audience, but we do. I think we all agree we want to electrify the grid. We want batteries, we want turbines, we want a whole host of green technologies, and almost every single one of those that has been proposed requires raw materials from mining. And at the same time, industry and governments are pouring billions of dollars into solutions for climate change. And so, yes, mining has an environmental impact, but we do need it. We need better solutions. Mining's also in the space where there's these large ore deposits that are harder to find. The ore qualities are decreasing. And of course, the social license and government policies have shifted towards more sustainable practices. And so we've got this need, and we've got this harder situation physically, chemically, geographically, socially. And so here we are. And so this is why mining needs microbes. Microbes can really help along the whole cycle of the life cycle of a mine, from exploration. So there was emerging evidence that bioprospecting might be quite effective. And so the idea would be drilling is expensive, especially in hard to reach environments, but microbes are a lot easier to get. If you can go up to a site and in the first couple of meters, things you could access with a shovel, take microbial samples, and from that get some kind of probability that there is the ore body that you want below there, you can then use that to narrow down your drillings. Maybe you have to drill less holes, for example. And as those models get better and better, they can become more and more accurate. There's also in the development, so we're seeing copper bioleaching in Chile representing about 30% of the copper that's produced there. There are passive bioreactors that are being used to treat acid rock drainage at the Lily Orphan Boy mine of State Run in Montana. The saturated rock fills for water treatment in the Elk Valley have been widely publicized, are in use, and are being quite successful. And then as the mine closes, of course, water treatment and tailings management become big areas where microbes can lend a hand. It's still hard to do. Individually, developing new microbial solutions can be expensive, and not all of them are working yet. There is a lot of work to do here. There's a high cost to identify individual microbes or group of microbes and genes that can perform the necessary function, and that's again because there's this huge diversity of microbes. We just haven't studied those microbes on mining sites very much. So we need to get that work done. We need those baselines, we need those databases built so that we can go and identify those genes and identify those microbes and figure out where they're living. There's also, it's complex, like I said, interactions with existing microbial communities and the environment in which they live mean that we have to tune the parameters. We need to understand how those changes in the environment affect the microbes so we can then manage them to do things that we want them to do. And the whole process requires a lot of collaboration and a lot of different areas of expertise. So you need miners and mining companies, you need academic institutions, you need solution providers, you need people that know engineering, that know microbial ecology, that know computer science to make this happen. It is a huge diversity of skills to really make this a thing. And so the project that I want to talk about here at the end is MMAP, so the Mining Microbiome Analytics platform. It is a consortium of mining companies, solution providers, and researchers, essentially, that have come together to build this platform. It's funded in part by the Canadian Digital Supercluster. They used to be called the Digital Technology Supercluster, but they dropped that part. It's just the Digital Supercluster. And MMAP is meant to be a library, sample analysis, and collaboration platform that accelerates the creation and development of microbial solutions. So we don't want these challenges to block the world from developing solutions. And we can do that by creating this platform. The platform benefits from having tens of thousands of samples to start. And so a lot of questions that we just can't answer today. How does season affect these microbes? Are the microbes similar between mining sites? How does different tailings affect the different microbes that are living there? How does weather affect the tailings if you're using some of the similar processes? How does time and how long it sits there affect the microbial community? Like, all of these questions are seemingly basic, and the platform will allow us to answer them. And that means that already you have a problem on your site, and we just know so much more than we did without the platform. So this quickly reduces development costs. You have a library of mining related sites and samples that you can go and look through and, no pun intended, mine. Improve performance. So the insight required to optimize the solutions in this field and is vast, since we need a lot of data. Like I said, it all comes back to these comparisons. DNA is not a blueprint, they are tools. DNA can be thought of as a tool set much more effectively. And so we need all of these samples to understand what are the different tools that these microbes are doing, and we need that data to compare new data to so that we can understand it really quickly. So we get improved performance by doing this. And so if you are able to process your microbial samples, your microbial DNA with a big database and platform like this, you will get more answers, more results, better answers. We call it annotation, so labeling who's there, what are they doing, who's doing what, basic questions like that. All of that will be improved by having a reference database to inform the answers to those questions. And so we really can give you more accurate and more complete information by using this platform than any other solution on the planet at this time. And it also allows for close collaboration. So sample owners maintain control of their data by using MMAP. You own your data, you control your data. There isn't a risk there because that confidentiality and data security is absolutely of utmost importance here. So there's a lot of work to do. Microbes have a lot of potential, and MMAP is really an exciting project to be a part of because I think it can help advance these technologies in a huge way. Below here are the logos of the companies that are already involved with MMAP. But as we expand into phase 2 and accelerate our development and go even further, we are always looking for more data, for more samples. You can join MMAP. We can look at your data. We can help you answer questions. We can talk to you about what might be realistic in terms of timelines and what you can learn from those samples. We're really here to collaborate with the community, tell everybody about MMAP and what we're building, and answer questions about how these technologies might apply to your own sites and your own challenges. My contact information is up here. And with that, we still have some time for questions.
Thank you, Aria. Thank you so much. Yes, we do have a few questions. So let's start with the first one. Okay, is it likely that surface microbes would be predictors for deep ore bodies? Would microbes proliferation linearly to surface, or would the path be nonlinear and dependent on nutrient resources, moisture content, and by avoid inhospitable subterranean biosystems?
Okay, lots of questions in there. Is it possible that surface microbes? Yes, it's possible. Actually, early evidence says they could be quite good predictors. So we're not talking about the microbes on the first couple of centimeters, but several meters down. So you really have to dig with that shovel, but could access it with a shovel. And the reason for that is that the underlying geological material can often be a good predictor of what's there. And so microbes are sensitive to small changes in those chemical and geological profiles. And so yes, it's very possible. And even likely, given, to be fair, small and initial pilot studies, but it looks good. It's not linear, no. Soils are really stratified, and then below soils, you have all this geological mass. You'll find microbes like deep down into crevices in subsurface environments. Very, very few of those are actually sterile. There are some exceptions. There's like paleosterilization actually is a concept. If you get enough heat and pressure for a while, there are places that can be sterile. But then as things cool or pressure decreases with depth, you actually see microbes kind of creep in there. All of those factors that you listed and more affect the microbial community, so you're absolutely on the right train. But there are other things as well, so that list is like very, very extensive.
Okay, and the same, you mentioned pilot studies, and the same person asked if you're running in, if you're doing field testing. Yeah,
so that's a great question. For bio exploration specifically.
Well, I think so, because it's the same person who has a previous question.
Yeah, fair enough. Okay, so generally, yes, all these samples are coming from the wild. We're doing field studies. We have use cases and projects, and we'd love to talk to people about doing that on their own sites as well. For bioexploration specifically, yes, we've done some pilots in the field. If you send me an email, I can dig up what's publicly available on that. It's not the whole set, but there is some information and data that is publicly available on that that could send your way. And we have a massive opportunity to collect a huge data set. And so this is a problem with AI and ML generally. You need a lot of data to build a good model, a lot of data. And so that's the best thing about being able to collaborate with mining companies, is that if this is something that particularly interests you, we can absolutely talk. And so if you're drilling, take a microbial sample. They're really easy to take, actually. You can be trained up on that pretty quickly. It's pretty inexpensive to process them for certain types of sequencing. And we can start to build that data set and make that model as robust as possible. So definitely an area of further exploration, but does look promising.
Okay. Then we have another question. So for the greatest benefit, is it a question of the right microbe in the right place, or a question of genetic engineering?
Oh, great question. So to be super clear, whenever you have a chemical or physical process that's working, keep doing it. As soon as you get biology in there, it's going to be a little bit tougher. It can be both. So the right microbe in the right place, yes. But I would argue, actually, we can tune and control that community a little bit. So even if we find that a microbe can degrade a compound partway, and then another microbe can degrade it the rest of the way, we could, without bioengineering, we could feed or starve that community with the right foods, that those microbes that can do the jobs we want. And so we can say, like, okay, we're going to stimulate this community, it's going to do this. Then we're going to stimulate this community, and it's going to do this. It's not like the right microbe in the right place makes it think like you really got to cross your fingers and hope that that guy's there. There are microbes everywhere. There's incredible diversity of microbes. There's incredible genetic repository and tool set almost everywhere. So there are many, many situations where you already have the right microbe in the right place. You just really need it to dominate the community to get that job done en masse. And so it would really be about changing the environmental parameters around it, adding molasses, adding ethanol, adding some kind of carbon source, restricting something that that made another population flourish in that environment. So yes, right microbe, right place, but they're probably already there. It's about right microbe making it the right place for them to really flourish. Bioengineering is absolutely on our radar as well. And so that's part of what the MAP platform will allow you to do, is say which microbes can do this process. So none. I don't really want to do it in 2 or 3 microbes. It seems like a lot of work. I'd like to bioengineer a microbe who could do it all together. So you'd be able to look through and find genomes that have many of the parts you're looking for. And you would be able to look at where on their genome, there are sites on a genome that are generally like easier to insert a gene there than other sites. So we'd be able to kind of show you some of those likely locations. It really is with microbes, if you get lucky, and this is more about luck and just trying a lot of things, but like the Matrix where they plug in and then they like upload new skills, you can kind of do that with microbes and bioengineering. And so that's another thing as well. It's harder than you think. It's harder than we all thought to, you can bioengineer this microbe and you're like, great. The tricky bit actually surprises people often, but if you want to take that bioengineered microbe and then put it into the environment, the tricky part is that the indigenous microbial population will usually just wipe it out. It's like, who are you? We've been here long before you. Like, no, they just kill it off. And so the bioengineering actually still needs that. I have to create the right environment for this new microbe to flourish there. So there's some additional challenges there, but can work really, really well.
Good, thank you. Another question, are there species overlap in which ore attracts which microbes?
Sorry, say that again.
Are there species overlap in which ores attracts which microbes?
Oh, like do we see the same species in different ore types? That is a fantastic
Yes, that's what
question.
I understand
Yeah,
too.
Yeah, that's a good question. So some of our preliminary analyses, and we hope to publish that in the coming months, actually shows that yes, there's species that overlap, but there are community signatures. So there's so many different species that would live in a single ore deposit that, like, yes, there's some overlap, which is good, thank goodness. We don't have to start fresh with every sample. But we can actually find signatures or fingerprints that are pretty specific to different sample types or different ore bodies.
Okay, and another question, are there any remediation studies active with microbes?
Yes, the oil science has been doing this for a long time. There's a ton of work up there for remediation. in tailings, in pits, all sorts of things, and many on mine sites as well. Some are public, some are private. It really depends on the way that study was set up and the company's comfort level with sharing that information. But yes, active around the world on all sorts of different contexts.
Okay, that's great. Well, it's the end of the questions list. So thank you everyone for joining us today. And if you have more questions or you would like to have more information on the MMAP project, you can contact Aria by email and she will gladly answer your questions.
Absolutely.
Thank you, everyone.